Hello and welcome everyone. I'm delighted to be here today as part of the PGC Worldwide Lab series of webinars. My name is Maria Niarchou and I'm a research assistant professor at the Department of Genetics Medicine at Vanderbilt University Medical Center. Today, I'm honored to chair the session titled PsycheMERGE Network, Using Electronic Health Records to Advance Psychiatric Genomics Research. We have a fantastic lineup of speakers who will share their exciting research with us. But before we begin, I'd like to provide a brief overview of the PsycheMERGE Consortium to ensure that we're all on the same page. The PsycheMERGE Network is co-led by Leah Davis, Jordan Smoller, and Chris Chabris. It was established in 2018 across 3 academic medical centers that all had EHR data available for research and linked biobanks with genotype samples. In just a few months, PsycheMERGE expanded rapidly and currently has 21 sites, including international partners. PsycheMERGE is built on the collaborative infrastructure of the eMERGE network and focuses on mental health and psychiatric conditions. It differs from the original eMERGE network in that it employs a federated analytic approach rather than a centralized data approach. This approach involves bringing scientists together to develop centralized pipelines that then get deployed locally, and the results are meta-analyzed across sites. So in total, the PsycheMERGE network now has partnerships across 21 academic medical centers, registries, and partner sites, over 80 collaborating members with access to over 15 million EHRs and over 950,000 genotype patients. So the PsycheMERGE network is divided into several work groups, and each work group has its own distinct focus. During today's presentations, we will have the opportunity to learn about the research conducted in 3 of these work groups. However, before we begin, I would like to make a quick housekeeping announcement. We will be conducting all the talks back to back, followed by a Q&A session at the end. So please feel free to post your questions in the Q&A, and we will have time to get to them at the end. So it's with great pleasure I'd like to introduce our speakers today, Drs. Roseann Peterson from SUNY Downstate Health Sciences University and Rachel Kember from the University of Pennsylvania, who are going to present to us some of their findings on behalf of the PsycheMERGE Diversity Initiative. Right. Stop sharing now.
Okay, great. I'll go ahead and share my screen. All right, all looks good?
Yes.
Okay, great. So thank you, Dr. Maria Niarchou, for the introduction to PsycheMERGE, and thank you to the PGC and Dr. Catherine Lewis for the invite to really showcase research from PsycheMERGE today. I'm Roseann Peterson. I'm from SUNY Downstate, and I'll be talking to you today on behalf of my co-chair, Dr. Rachel Kember of Penn, and the PsycheMERGE Diversity Initiative. Rachel is here today and will also help facilitate discussion on this work during the Q&A at the end of the session here. So here we are taking a deeper look at our psychiatric phenotyping from electronic health records. And as an example today, we'll be looking at a bipolar disorder algorithm. Okay, here's my disclosure slide. No conflicts of interest to report, but this work is supported by the U.S. National Institute of Mental Health, as well as the Brain and Behavior Research Foundation. We'd like to thank them for their support. So why do we need a PsycheMERGE Diversity Initiative? A disproportionate majority of participants in our genetic studies are of European descent. This left box here, in general, shows the proportion of major world populations: African, East Asian, European, Hispanic, Middle Eastern, Oceanic, and South Asian. And the right boxes show the representation we have in our genetic studies of schizophrenia, of bipolar disorder, and major depression. So as you can see, our genetic studies are not reflective of global diversity, and so we must continue our efforts to reach sample size parity. Which brings me to the PsycheMERGE Diversity Initiative, which is one such effort. Electronic health records, or EHRs, they're collected in real-world clinical settings, makes them particularly available for research aimed at reflecting population diversity. So we formed the PsycheMERGE Diversity Initiative, which is a dedicated effort to increase representation in our genetic studies of psychiatric disorders by really leveraging EHR biobanks. And currently the initiative consists of data from 6 sites. We have Mount Sinai, BioMe, Vanderbilt BioView, Geisinger Health System, Mass General, Million Veteran Program, and Penn Medicine Biobank. So the initial goal of this initiative was to improve ancestral representation across GWAS studies of psychiatric conditions and imply inclusive trans ancestry genetic approaches. But we recognized as a first step that we had to ensure quality and equitable psychiatric phenotyping across groups. And so assigning case status is an important step in conducting psychiatric research using EHRs. However, it is often defined using phenotypic algorithms, which have been developed for majority white, non-Hispanic individuals. And bias in assigning these diagnosis codes across groups could lead to poor performance and exacerbate health disparities in our research. So here in the PsycheMERGE Diversity Initiative, we are investigating these issues by examining prevalence differences in case assignments, and then differences in prevalence of features and counts of these features that are used in the psychiatric algorithms across biobanks and across EHR reported race and ethnicity. So the purpose of this work was to examine if there were differences in bipolar disorder case assignment determined by the algorithm across EHR reported race and ethnicity, and then if this was driven by differences in prevalence and counts of specific EHR attributes or features contained in the algorithm. So in order to examine differences across the 6 diversity initiative sites, we selected an already developed bipolar disorder algorithm. And I know not everyone is familiar with EHR algorithm development, so I'll just give a bit of background on the bipolar disorder algorithm that was applied across the 6 biobanks. So in brief, the algorithm was developed and validated against clinical chart review by the team at Mass General Brigham as part of the parent eMERGE 3 network, using a method that combines coded features in the EHR, such as ICD codes and medications, and then applying a machine learning logistic regression framework. So what does that entail? First, data are filtered to identify subjects with at least one ICD diagnosis code for bipolar disorder, who also met the predefined data floor. Okay, so what is the data floor here? It was defined as having 3 or more diagnosis codes, right, any codes, on 3 separate days and at least 1 clinical note. And this is termed the screen positive set. Next, chart reviews were then performed on a random set of 200 charts selected from the screen positive set to establish a gold standard training set. Then domain experts performed the chart reviews using the International Cohort Collection for Bipolar Disorder and Diagnostician Review for Diagnosis and Confidence. Then these chart review results were used to train the machine learning regression model. So a set of EHR attributes, also called features, associated with bipolar disorder were defined using surrogate assisted feature extraction, also known as SAFE. SAFE advances high throughput phenotyping methods by automatically selecting a set of informative features for the algorithm training. And then these potential predictors are included from 5 publicly available knowledge sources, including Medscape, Mayo Clinic Diseases, and Medline, which then yields a unified medical language system set of concepts as the candidate features going into the machine learning. Okay, penalized logistic regression model LASSO was trained to classify the bipolar disorder in the gold standard training set. The regression model then identified the relative weights or the beta coefficients of each feature significantly associated with bipolar disorder diagnosis. Okay, so once the model was created, a threshold value based on a specificity of 95% and a positive predictive value was selected to identify bipolar disorder cases, right? So these are those above the cutoff are identified as cases, and those below are non-cases. Okay, so displayed here are the results of machine learning logistic regression model. You can see the features are over here on the left and the regression weights here on the right. And there are features related to bipolar disorder, hypothyroidism, depression, suicide, as well as medications. And the predicted value for each individual is calculated by the count of each feature multiplied by the regression weight. And so each feature or concept in the model is made up of related ICD codes. And here showing the codes included for hypothyroidism. And here's an example of the list that went into the medication features for valproic acid. Okay, so I've just shown how the algorithm was developed and the corresponding features and weights in the algorithm. Now, there are also validation steps. Okay, so the algorithm was then implemented in an additional 200 individuals with expert chart reviews. So this is our truth set in order to estimate the positive predictive rate of the algorithm, comparing the chart review results right to the algorithm results. So here, this algorithm reached 89% positive predictive value, which is the rate of the true positive in those classified as cases by the algorithm. Okay, so we've just gone through the steps of how the algorithm was developed and validated. Now recall that this algorithm was developed without consideration to demographics like EHR reported race and ethnicity. So from our perspective, from the Diversity Initiative, we wanted to inspect the representation of the individuals that went into the training and the validation data sets. Okay, so here's the table. Oops. Sorry about that. Okay, here's the table of the EHR reported race of the training set and the validation set used to develop this bipolar disorder algorithm. And as you can see, it was trained and validated on a majority white non-Hispanic, about 76%, followed by Black non-Hispanic individuals, 6 to 8%. So this is not representative of the US, but this is largely reflective of the demographics of the biobank as a whole. Okay, so now inspecting how the algorithm performed in white, non-Hispanic, and non-white participants, the validation data set positive predictive value and the sensitivity point estimates here are indices of the true positive negative rates were lower in the non-white group. Okay, so the PsycheMERGE Diversity Initiative as a first step would like to assess if this influences performance of the algorithm across groups and across our 6 biobanks. So now that we have the background on the bipolar disorder algorithm, here in the current study, we wanted to implement this algorithm across our 6 PsycheMERGE Diversity Initiative sites, and then examine if we see differences, not only in case status determined by the algorithm, but also why. Is this due to differences in feature prevalence or counts of the features contained in the algorithm? Also, as an aside, I'd like to note that race and ethnicity in the EHR were either self-reported or reported by medical staff, which is why we're referring to this as EHR reported race and EHR reported ethnicity. Okay, so the analytic plan, each site implemented the bipolar disorder algorithm and determined case status stratified by EHR reported race and ethnicity. They further calculated the prevalence and count for each feature in the algorithm by EHR reported race and ethnicity. And then the results were meta-analyzed in our meta package using a random effects meta-analysis due to known heterogeneity across biobanks. So the EHR reported race and ethnic groups that had data available at 4 or more sites were analyzed here. Okay, so here are the sample sizes for each PsycheMERGE Diversity Initiative site by EHR reported race and ethnicity. About 1.5 million individuals in total, 1 million are white non-Hispanic, 200,000 Black non-Hispanic, and close to 50,000 white Hispanic. So the bipolar disorder algorithm was implemented at each site, and here are the results of the percentage of individuals who met the predicted probability threshold to be defined as a bipolar case. The overall bipolar disorder prevalence rate was 1.06% and ranged from a low of 0.6% in the Asian group to 1.27% in the Black non-Hispanic group. Okay, now to identify reasons for these differences, we explored both the prevalence, the percentage of individuals receiving at least one code, and the count, the number of codes received for each of the features in the algorithm stratified by EHR reported race and ethnicity. So this figure is showing the percentage of individuals with one or more codes for each feature in the bipolar disorder algorithm. And the mean percentage for each strata were calculated at each biobank site, and then the results were meta-analyzed across the 6 sites. On the y-axis here, we have each feature that went into the algorithm, and the bars represent different EHR reported races and ethnicities. Okay, so 3 of the 7 features in the algorithm showed significant differences by EHR reported race. The percentage of Black individuals assigned an ICD code for bipolar disorder is more than twice the percentage of Asian individuals. Now for hypothyroidism, the white group had higher rates of hypothyroidism than the Asian or the Black and Black Hispanic individuals. And for depression, the Asian group had significantly lower percentage of depression codes than the Black individuals. Okay, so next we looked at the count, that is the number of codes received for each of the features in the algorithm stratified by EHR reported race. Individuals with one or more code for each feature were analyzed. Again, mean counts for each EHR reported race and ethnicity group were calculated at each biobank site, and then results were meta-analyzed across the biobanks. And there were no statistically significant differences for feature counts, but we would like to note that although we saw the percentage of Black individuals assigned an ICD code for bipolar disorder is more than twice the percentage of Asian individuals, among those that have at least one ICD code for bipolar disorder, the average number of codes is twice as high for Asian individuals compared to Black or white individuals, which is shown here. And this matters, right? Because the algorithm is weighted by the count, how many times someone has a code. Okay, so in summary, previous bipolar disorder algorithms have been developed without regard to race and ethnicity and predominantly trained on white individuals. We applied the bipolar disorder algorithm previously developed by the eMERGE3 network to 6 PsycheMERGE biobanks, and we saw significant differences in the performance across EHR reported race and ethnicity, specifically the proportion identified as cases and feature prevalence of bipolar disorder, hypothyroidism, and major depression. Okay, so EHRs represent complex, multi-level human decisions that encode several levels of systemic bias in arriving at treatments, at diagnosis. So differences in phenotypic and diagnostic accuracy between groups may occur. In the PsycheMERGE Diversity Initiative, we are working towards inclusive algorithms that are trained and validated in gold standard sets that are representative to really better identify individuals with bipolar disorder, schizophrenia, and major depressive disorder from these complex EHR data. So really, in order to improve algorithm performance for all, which will in turn facilitate our inclusive genetic studies. Okay, so this figure on the right, the top panel shows the general representation of the world and the US. Again, we have African, East Asian, European, Hispanic, Middle Eastern, Oceanic, and South Asian. And the left panel is where we are today in our representation of published GWAS studies. So you've already seen this slide today. But the right panel is where we will be after we have included data from the PsycheMERGE Diversity Initiative. My cat wants to say hi. So you can see the samples are adding up, and while we still have a ways to go to reach sample size parity, we can make a difference by aggregating across our EHRs. Okay, so with that, I'll end there, and I'd like to thank all the Diversity Initiative members and their teams. Also, if you're interested in collaborating, please let us know. I'll also do a real quick shameless plug. If you're interested in this type of work, trans ancestry genetic analyses, EHR phenotyping, myself and co-chair Rachel Kember and other PsycheMERGE members are hiring. We're hiring postdocs, data scientists, programmers, project managers. Please reach out and spread the word. And I'll stop there. Thank you for your attention, and looking forward to discussing this further in the Q&A session at the end of the talks. Thanks.
Thank you so much, Dr. Peterson, for sharing this important work. Again, if you all have questions for Dr. Peterson or Dr. Kember, please feel free to add these to the Q&A, and we'll get back to them at the end. So our next speaker is Dr. Karmel Choi from Harvard Medical School, who is going to talk to us about some of the work from the PTSD work group.
Thanks, Maria. And thanks, Rosie, for that great talk. Hi, everyone. It's great to be part of this worldwide lab that's showcasing the PsycheMERGE consortium with such amazing groups and colleagues. So thanks again to the PGC for the opportunity and to Maria for bringing us together as well. As Maria mentioned, I'm Karmel Choi, and I'm a clinical psychologist and assistant professor at Harvard MGH, where I direct the Precision Prevention Program within the MGH Center for Precision Psychiatry. And today I'm presenting on behalf of the PTSD Trauma EHR Working Group that's hosted by the PsycheMERGE and PGC PTSD. It's been estimated that 90% of all adults in the United States will experience at least one traumatic, potentially traumatic event in their lifetime. And so this puts many of us, almost all of us, at risk for developing some type of post-traumatic stress condition, of which PTSD is one of the most debilitating. We know that PTSD is associated with high levels of distress and disability. Not only that, it is also increasingly recognized as a contributor to costly health problems, such as cardiovascular disease, and more recently, autoimmune conditions, GI pain, etc. And while these specific disease associations have been identified in the literature, what has remained lacking is a more comprehensive understanding of the full range of medical conditions. In other words, the clinical phenome that is both known and not yet known to be associated with PTSD. And this knowledge is important as it could inform care allocation, being able to anticipate what other healthcare needs that someone with PTSD may also present with. It can guide prevention efforts, the identification of health conditions that we might want to treat in advance to reduce future PTSD risk, or health conditions that may need to be averted among people living with PTSD. And then finally, this knowledge could also spur further and potentially novel research into the clinical, genetic, and other mechanisms that link PTSD to these common health conditions. So to paint this type of comprehensive picture, we have this opportunity to draw on large-scale EHRs from healthcare settings, which is obviously the focus of today. And one major consideration here, as Rosie has already mentioned, is the topic of phenotyping, how we actually can identify PTSD cases from EHRs. So PTSD can be defined through a specific set of diagnostic billing codes. However, providers may often choose to assign broader codes in the course of treatment, which means that some PTSD cases may actually be missed when we simply focus on a very narrow set of codes. And so if we expand to a broader set of stress-related codes, this may help us to capture more potential PTSD cases, which enhances our statistical signal. However, these more minimal phenotyping approaches may also introduce non-specific associations, as Na and others have shown in the case of major depression. So how we identify individuals with PTSD from our EHRs may certainly impact our downstream research, and we need to start to characterize how. Along the way, it is important and quite relevant to assess sex differences in these patterns, especially because PTSD burden differs markedly by sex, with females showing almost twice the lifetime prevalence of PTSD compared to males. And so we sought to address the following research questions broadly: which medical or clinical comorbidities are most strongly associated with PTSD in major EHR systems? To what extent does this vary by how PTSD is defined operationally? And do these patterns of association vary by sex, that is, in male versus female patients? And so here we drew upon the PsycheMERGE Network led by Jordan, Leah, and Chris, which gave us the chance to look across multiple health systems to examine the EHR-wide clinical correlates of different PTSD definitions. And this effort also stemmed from a collaboration between PsycheMERGE and the PGC PTSD. So it was supported in part by an administrative supplement to the PGC PTSD R01 led by Caroline Nievergelt, Kirsten Conan, Murray Steen, and Carrie Ressler that was specifically focused on bringing in EHR data sources to increase sample sizes for GWAS, for which I served as the site PI. So for this study, we drew on EHR sites from 3 major healthcare systems, Mass General Brigham, the Vanderbilt University Medical Center, and Mount Sinai through their biobank, BioMe. And before moving on, I want to acknowledge the analysts and collaborators who've directly worked at these sites on this particular project. Initially, Slavik Guleva from Leah's group at Vanderbilt, and then also Maria Niarchou from Vanderbilt, Emily Hicks from Laura Huckins' group at Mount Sinai, who has now been driving the paper as first author and has produced much of what you'll see in the most updated results and figures. And then, of course, Laura, who's been fantastic in collaborating and also guiding this work as well. So it's been a really great group effort to harmonize our approaches across sites and learn from each other in this initial effort. So we had a combined sample size of about 123,000 biobanked patients across 3 health systems. And all sites had relatively similar mean age. They were about 55 to 60% female in composition and had a range of genetically determined ancestries. We focused on 2 definitions of PTSD that had actually been considered for the PGC PTSD GWAS freeze. So the first definition was a narrow definition of PTSD in which cases were individuals who had 2 or more PTSD-specific codes, while the controls were individuals with no such codes and also no post-concussive disorder codes, no stress-related disorder codes, so fairly clean as controls. On the other hand, we had also a broader-based definition of PTSD in which cases were those who had 1 or more PTSD or stress-related disorder codes, while controls consisted of everyone else. So here, narrow PTSD, as we might expect, picked up about a smaller proportion of 2 to 4% of the sample at each site. On the other hand, broad PTSD picked up more cases, 7 to 22% of the sample, with particularly high numbers in the MGB biobank, which does tend to be overrepresented for common psychiatric conditions. And to evaluate the clinical conditions that might be associated with each of these definitions, we drew upon the Phenome-wide Association, or PheWAS framework. And a PheWAS relies on Phe codes as the outcomes or endpoints. And so these are really disease groupings that help to provide some dimension reduction for the large number of ICD codes that are otherwise floating around in the medical system. So there are about 81,000 different ICD-9 or 10 codes that one can receive at any point, but they can actually roll up into about 1,800 distinct Phe codes in total. So more parsimonious. So we conducted the PheWAS using logistic regression models. We examined the association of PTSD case status in relation to each Phe code, adjusting for covariates. And then we ran similar stratified models in male versus female patients. And then finally, we examined for Phe codes that were associated with PTSD in either the full models or the sex stratified models. We tested formally an interaction term between PTSD status and sex in relation to those Phe codes. The PheWAS analyses were conducted at each site, and where applicable, the results were combined across sites using meta-analysis. And briefly, although not the primary focus of this study or my talk today, we briefly did examine the association of each of these definitions with a PRS that's trained on the latest published PTSD GWAS, which contains mostly clinically ascertained cohorts. Just as we're mostly looking at the clinical correlates, but to perform a little bit of a genetic validation of these 2 different definitions. And I'll jump into that actually first before getting into the main results, just so we can see that. We basically see at the top in the European subsample that there is a slight difference in the betas associated with narrow versus broad PTSD in relation to the clinically ascertained PTSD PRS. And similar patterns although less statistically powered in the African ancestry subsample, of which only 2 sites had available data. But the basic takeaway message is that we can see some differences across the 2 definitions at the genetic level. And now we will turn to looking at more of the clinical phenome differences. So here is a side-by-side comparison of the overall PWAS results. So narrow PTSD on the left and broad PTSD on the right. And all the possible associated phenotypes are arranged vertically along the y-axis by disease type, with the log p-value shown on the x-axis going out. So here we can see the narrow PTSD definition indeed generates a pattern of comorbidities that is relatively narrow. So it's particularly enriched, as we can see in the mental disorders category, and this encompasses anxiety disorders, substance use disorders, severe mental illness, personality disorder, etc. And so while that does seem to be the major peak in the association results, there is also a slight elevation of somatic conditions as well near the top that included things like back pain and abdominal pain and other somatic states. On the other hand, the broad PTSD definition has a pattern of comorbidities that we can see is a bit more spread out across all the disease categories. Certainly elevations in association in the mental disorders domain, as well as the pain and somatic conditions, but also a range of conditions, including respiratory diseases, GI conditions, genitourinary conditions. So it's further spread out across the phenome. And reassuringly, most phe codes were actually, in fact, shared across both broad and narrow definitions of PTSD. So this graph is basically plotting the significant, Bonferroni significant hits in terms of their p-value of association for broad PTSD on the x-axis and narrow PTSD on the y-axis. So in the purple cloud of dots are things that were associated strongly in both. And so we can see this cloud largely consists of a lot of psychiatric phenotypes, but there was also quite a few, if you look at the red cloud of dots down below, that were specifically associated with the broad definition of PTSD, but not with the narrow definition. And so it's kind of consistent with what we eyeballed before in the PWAS plot, but we can see it sort of in a different way here, with some of the most strongly specific associations being things like in the cancer or neoplasms category. And interestingly, the phe code associations with PTSD were primarily positive, which means that PTSD status was largely associated with increased risk of also being seen for these conditions as well. And so there was only a very small number of phe codes that were negatively associated, in which having PTSD meant that you were less likely to be seen as having these other conditions. And this included phenotypes such as enteritis, so the Crohn's disease basically, as well as prostate cancer. And these seem to be fairly robust because the direction of effect and as well as the significance were also seen at each of the biobanks. And so we have been discussing this a bit more in terms of why that might be the case, but if folks have any thoughts or suggestions, we are also very much open to helping with the interpretation for that. So looking at the sex differences results, there was nothing particularly robust in the narrow PTSD definition, so I won't spend time there today, but here in the broad definition, I'm showing here an interesting plot that Emily generated, and it's clever because it shows the sex by PTSD interaction betas for each of the sites. And at the top of each cluster of dots is the meta-analyzed beta in red. And so for today, what you basically want to focus on is simply which side of the beta equals 0 line that the meta-analyzed beta falls. So if it falls to the left of the line, it suggests that the association with PTSD is enriched in female patients. And if it falls to the right of the dotted beta equals 0 line, it means that the association with PTSD is enriched in male patients. So I'll run through some of those so you can take a look. So in terms of associations that were stronger in males, this included osteoporotic conditions, respiratory failure, different types of brain hemorrhage, pulmonary heart disease. And so those were the main ones that seem to be enriched in association in male patients. And then in terms of stronger associations with PTSD in females, this included urinary tract conditions, acute pharyngitis or respiratory infections, which basically means sore throat or infection. And then also the phenotype of overweight or obesity. And so broadly, what the takeaway here is that PTSD, especially when broadly defined, may co-occur with different health conditions that vary between male versus female patients, although in general, their overlaps in terms of associations are similar. Okay, so that was a whirlwind just preview of what we've been putting together, but the summary that you can take away is that there are certainly many substantial associations between PTSD and the clinical phenome across 3 major healthcare systems. We saw that definition matters, where broader EHR-based definitions capture more comorbidity with a range of somatic conditions. And then we also saw some sex differences in the conditions that were associated with PTSD. Many shared comorbidities, but also some specific conditions, some more severe kind of heart, lung, or brain related conditions in males, and then urinary system conditions and metabolic and infection conditions in females. And largely this work, as with many sort of PWAS or GWAS type work, is hypothesis generating. And so this raises some questions about the potential mechanisms of these co-occurrences or associations. So are some of these health conditions true risk factors for PTSD? Are they a consequence of PTSD? Or could they be simply a manifestation of how and where PTSD gets diagnosed? And so where you might just be detected as having that PTSD or a co-occurring condition, or can it be related to treatments that someone has received as well? But in any case, it does highlight the importance in all of our work for choosing our definition, our EHR-based definition for GWAS and other studies of PTSD and psychiatric conditions within these types of healthcare settings. And so the broad takeaway is that this work certainly identifies known as well as novel PTSD comorbidities across multiple health systems and sex differences that can be further explored for their clinical and genetic mechanisms. And so just to preview some of our next steps, we're wrapping up this current effort. And then there is also, so this was more of a phenotype PWAS, and there's also a PRS PWAS led by Gita Pathak, that's also forthcoming. But some of the work that we hope to do moving forward is to contribute more diverse, as Rosie mentioned, cases and controls for GWAS. Also doing work to model EHR data with genomics and other omics. And then also we're actually currently in the stage of brainstorming these further directions and collaborations. So if you have the data and the interest, please do join us. It would be interesting to think of things that are also maybe kind of spanning related conditions that were identified here in this PWAS. So with that, I wanted to thank the working group that has been involved, which spans these various groups here, as well as the PGC, PTSD, PsycheMERGE, with a special thanks to Emily Hicks, who has really been driving this project that I presented forward from Laura Huggins' lab. So thank you all, and I look forward to discussing alongside my colleagues in the Q&A.
Wonderful. Thank you very much, Karmel. So our next speaker now is Dr. Sandra Sanchez-Roige. He's going to present some very exciting findings, looking at doing a multi-instrument analysis of tobacco use disorder.
Thank you, Maria. It is an absolute pleasure to be here today. My talk has 2 threads. The first one will argue the value of electronic health records to accelerating discovery for tobacco use disorder. The second thread will argue or showcase the benefits of working in an extremely collaborative network such as PsycheMERGE. Admittedly, we weren't sure when we began the journey what the quality of the phenotype would be, but I hope that by the end of the presentation, you will be as surprised and pleased as we were with the set of results that you're about to see. The work that follows is the result of the many bright smiles that we can see on these slides from the members of the Substance Use Disorder Work Group that I am fortunate enough to co-chair with Dr. Vanessa Troiani. And we're all united with the same goal of unraveling the biology underlying substance, and particularly for today, tobacco use disorder. But their etiology is complex because they develop in a multi-stage process, from initiation to regular use to harmful compulsive use, cessation, relapse. And it is possible that different genes may act or have a different role at different stages. And for initial stages of vulnerability, such as aspects of smoking initiation and consumption, GWAS have uncovered hundreds of associations, as shown in this landmark paper by the GSCAN Consortium in 2018, and further saturating the number of loci by using millions of diverse individuals in a more recent study. But later or more severe stages of vulnerability, such as tobacco use disorder, they have been largely unexplored, which is perhaps unsurprising because ascertaining high volumes of cases is expensive, slow, requiring clinicians to establish a diagnosis using the gold standard DSM Manual of Mental Disorders. In 2020, our colleagues from the Indigo Consortium took a different approach and published another influential paper looking at nicotine dependence via 6-item self-reported questionnaire, as I shown in the slides, commonly referred to as FTND. And despite all these terrific undertakings, it was clear that larger and more diverse samples would be needed to move the field forward. Over recent years, as Maria and the previous speakers beautifully illustrated, there has been a shift in the field in which electronic health records, or EHR, can be paired with genomic information to fuel genetic studies of mental health traits, which is, of course, the premise of the PsycheMERGE Consortium. So we began with a clear goal in mind. Can we prove, or disprove, that we can use EHR information to identify cases and controls for a trait that is otherwise hard to collect at large scale, such as tobacco use disorder diagnosis? We studied a very simple phenotype that can be extracted from EHR, whether someone had 2 or more diagnostic ICD codes for tobacco use disorder, more commonly known as PHI codes, as Karmel illustrated. These codes may be derived by psychiatrists, but they may also be derived simply for billing purposes, so they are imperfect in nature. And we wondered if we can show that these traits, which can be considered minimal phenotyping, if we can show that it captures signal that is relevant to tobacco use disorder, we could show that it could serve as a cost-effective strategy for genetic studies of tobacco use disorder. So we gathered data across 4 sites within the PsycheMERGE network, across multiple ancestries, and just within 4 months, which is remarkable and one of the strengths of working in this consortium, Mariela Jennings, Sylvanus Stoikomo, and Hanjun Lee performed GWASs of tobacco use disorder PHI codes at each of the sites. Reassuringly, tobacco use disorder PHI codes were genetically correlated across sites, which encouraged us to meta-analyze the data. And here are the results for each of the 3 ancestries that we studied, overlaid on top of each other using different colors. We identified 55 loci in the European cohort in red, 2 loci in the African cohort in blue, and despite our attempts at increasing diversity, we did not identify any significant associations in the relatively smaller Latin American cohort. So to boost power, we next performed a multi-ancestry meta-analysis, which was led by Sylvanus Stoikomo, and we were positively surprised by the number of loci that surpassed the significant threshold line. And here was our turning point, when we realized that all the GWAS loci that our analysis identified replicated previously known associations with smoking phenotypes, from initiation to consumption, cessation, dependence, which was the clearest evidence of the value captured by tobacco use disorder PHI codes. This was also reflected not only by the quantity, but also the quality of the signals, as you will see in the next 4 slides. First, nicotine receptor genes, which of course need no introduction. Second, all drugs of abuse activate the mesolimbic dopamine reward pathway, and we can see here several dopamine-related genes were associated with tobacco use disorder. Third, substance use disorders have been speculated to be a disorder of learning and memory, and we found associations with variants in a gene that encodes for a specific glutamate receptor, and glutamate is, of course, an important neurotransmitter for mediating reward-related learning and memory. And similarly, we identified variants in the gene BDNF, which is also involved in memory consolidation processes. We then aggregated all these variants, even those that did not reach genome-wide significance, and we mapped them onto genes via various methods that will be familiar to many of us in this course. So you will allow me, for the simplicity of my descriptions in the interest of time, either positionally, via cis-regulated gene expression in various brain regions, chromatin, and protein abundance. And these various methods uncovered the striking number of 330 candidate risk genes for tobacco use disorder, 75 of which were not identified in prior smoking GWAS, which can, of course, now open up novel hypotheses to be tested experimentally. In aggregate, these genes were enriched in nicotinic and dopamine-related pathways, as well as enriched in brain tissues, including regions such as the frontal cortex, limbic system, which have been studied for decades in the field of addiction. In this busy plot, we also learned that tobacco use disorder risk genes, here squished on the right, showed interactions with varenicline, as we expected to find because it is the FDA-approved drug for smoking cessation, as well as with other compounds that seem to mirror conditions that are highly prevalent with tobacco use disorder, such as the target of antipsychotics, antidepressants, and dopaminergic agents. Tobacco use disorder was also strongly genetically correlated with other comorbid conditions, particularly tobacco and other substance use traits, as we expected, here clustering on the upper left corner, as well as this audience will be interested to see, other psychiatric and even medical conditions. We then extended this analysis, which included datasets from public GWAS, to another site from the PsycheMERGE Network, the Mayo Clinic, in collaboration with Dr. Brandon Coombs and Jana Biernaka. And for each individual in this biobank, we calculated polygenic scores for tobacco use disorder, and we performed a phenome-wide association study of PWAS against thousands of other medical traits. Tobacco use disorder polygenic scores were positively associated with tobacco use disorder, as defined via FICOTS in this independent site, which served as a positive control, as well as a plethora of other conditions, from other psychiatric and substance use disorders to metabolic, respiratory conditions, pain, among many, many others. We also extended this analysis to a deeply characterized sample recruited for genetic studies of specifically substance use disorders, the YALPAN sample. And again, for each individual in this cohort, we calculated polygenic scores for tobacco use disorder, and we performed a PWAS, in this case against hundreds of other traits derived from a comprehensive psychiatric interview, the SADA. And we were extremely pleased to see that our traits, again defined via EHR, ICD codes or FICODES, was strongly associated with many substance use traits, including nicotine dependence, as defined via psychiatric diagnosis, here highlighted in yellow, in both the European cohort, as well as in the African YALPAN cohort. At this point, some of you may rightly be thinking, how dissimilar, really, are these results from those of prior nicotine-dependent GWAS, such as those of FTND, as you may recall from an earlier slide. When we indeed compared our results, which are here as red dots, from those of FTND, here in gray, we indeed identified that tobacco use disorder polygenic scores were more strongly associated across virtually all domains, including nicotine dependence, than FTND polygenic scores were. again, emphasizing the value of deconstructing information or collecting information on later stages of vulnerability or more severe phenotypes, such as tobacco use disorder diagnosis. And lastly, we extended our polygenic analysis to a younger population, the adolescent brain cognitive development or ABCD cohort. Luckily, substances could not be captured in this very young population, at least at the particular wave that we studied in children of 9 to 11 years of age. But externalizing behaviors, which emerge in childhood and are strong correlates of substance use, they can be measured. And with the help of Emma Johnson and Alex Khatum, we found that tobacco use disorder polygenic scores were strongly associated with externalizing, here at the very top, as well as internalizing cognitive and weight-related phenotypes. And I'd like to emphasize that these children have not been exposed to tobacco. So we may speculate that any of the associations that we observed may not be a consequence of smoking, but rather they may underlie overlapping genetic architecture across the traits that we studied. So overall, it is undeniable, after scrutinizing the data, the quantity and quality of GWAS signals in diverse populations, the enrichment in relevant pathways and tissues, the genetic overlap with nicotine-related traits from deeply characterized samples, they all provide a proof of principle that EHR, in particular ICD codes, can serve as complementary tools to study tobacco use disorder genetics in a cost-effective manner. And because polygenic analysis revealed that tobacco use disorder is overlapping, as well as clearly distinguishable from other smoking phenotypes, we are now turning our attention to multivariate analysis to combine our data with these other prior smoking GWAS, including ongoing efforts by the PGC Substance Use Disorder Workgroup to better understand the full spectrum of smoking behaviors and their relationships with other mental and even physical conditions. This work is truly collaborative. I list the funding sources, biobanks, and again show the smiles of the many researchers that have fueled this project and many more that I was unable to fit in a single slide, with a loud shout out to Rachel Kember and the core group of talented analysts, Silvana Sikomo, Mariella Jennings, Hyunjun Lee, and Benjamin Pham. And of course, I must end by shining back again the spotlight to the PsycheMERGE Consortium and the inspiring leadership of Leah Davis, Jordan Smoller, Chris Chabris, without whom none of this would have been possible. This is my final slide. There is much more than we hope to accomplish, and I can elaborate more in the Q&A section. This work is available on MediArchive if anyone is interested to read a more comprehensive version of the slides. It is fair to say, on behalf of the group, we look forward to hearing all your ideas and feedback to further enrich the projects that we have presented today and foster collaborations. And I am very glad that we have extra minutes to do exactly that. Thank you.
Great. Thank you so much, Sandra, and to all our presenters. So far, we don't really have any questions, so you've all done an excellent job. If you would like to ask a question, you can either use the Q&A or you can raise your hand, but we can get the discussion started. Perhaps with Sandra, if you could also talk to us a little bit about the future directions with your project, as well as if you can tell us any other projects that may be interesting from the SUD workgroup, PsycheMERGE workgroup.
Thank you, Maria. One thing that we learned was that, as we expected, the trait is very loud, it's messy, and there's misclassification. And despite the noisiness, if you will, of this phenotype, it is, as we've seen, undeniable of the quality of the signal that we've just seen. But we want to do more. We can generate more phenotypes by leveraging on one of the strengths of EHR, which is we can use multiple data modalities, not only ICD codes, but we can also extract data from notes. And there's beautiful work by other members of the group, such as Alvin Jeffrey, for instance, and many, many others that are working on tools to make it easy for researchers interested in genetics to be able to use data from the notes. From that, we can also generate phenotypes that will be based on longitudinal data, since most GWAS tend to concentrate on static phenotypes that have been measured in adults. By having access to electronic health records since birth to very later on the lifespan, we can begin to unpack what are the age effects that we're seeing, especially considering that some of the psychiatric disorders that we're interested in studying have an early onset. And how does this vulnerability change and manifest across the lifespan is something that we really are turning our attention to.
Yeah, that would be fantastic. And speaking of like algorithms and like how we can define them, from work from the alcohol use disorder and child reviewing and the opioid use disorder with Dr. Jeffrey, we do find that we could potentially have sex-specific algorithms just because it looks like there are differences in how they present in the EHR. And I'm wondering, in terms of the race-related algorithms, for, a question for Rachel and Rosie, are you thinking of integrating the sex-specific aspect as well?
That's a great question. It's not something we've looked at yet, but our hope in our algorithms was actually, rather than doing different algorithms within each race, is that we could create a better algorithm that would work for all races and ethnicities, in part because we wanted to perform the genetic analyses in a trans-ancestry fashion without first splitting by ancestry. And so, we really want to be able to include individuals that are admixed and so on, rather than throwing them out of analyses. So I think there is a conversation in the algorithm field to be had at the moment about whether you do sort of define groups first and get algorithms that may be more accurate within groups, but require you to define a group, or whether you can find an algorithm that actually works for all.
Yeah, so just to add on to what Rachel said, I think the idea is to move towards using this federated learning model, so that that we're not just tuning on one or two demographics, but we can tune on multiple demographics so that we can improve algorithm performance in all.
Thank you. Here's a question for Roseann. In the Q&A, it says that we've seen differences in prevalence for various disorders in different ancestry groups for decades, but it is so hard to interpret them. How can we sort through the various explanations?
Oops, apologies, I was muted. Rachel, do you want to take a stab, or should I go ahead?
You go ahead.
Okay. Right. So I think I know as geneticists, we're sort of focused on, genetic differences and this and that. But I think we all know that our psychiatric phenotypes have large environmental factors. So I think we need to start incorporating these important environmental factors into our models. And I think that is going to help parse what's due to genetic effects, what's due to social determinants of health, and things like that.
I think we have 1 more minute. I don't see any hands raised or any other questions. So I have one more for Karmel, and it's about the sex differences. I'm wondering if which would be the ones you expected, and which ones would be the ones that were novel to you? And I also see we have one hand raised.
Yeah, well, I'll try to get through that quickly just to address, and I think similar to like maybe answers to your differences, it could be sort of like endogenous sort of vulnerability, but also like environmental and sort of human. And so one of the ones that we have discussed in our group, and as you would may remember, is the urinary system conditions. And so some of those were enriched in association with PTSD in female patients. And so that one, we wondered if that might be actually sort of an artifact of sort of healthcare treatment. So like if someone is presenting with a urinary UTI or like a urinary system infection, then it's possible that the conversation around sort of prior trauma history might also be kind of related to that. So the possibility of assessment may be more likely kind of in certain sort of healthcare settings. And so that was one that I think maybe we could think of a particular context in which that association could emerge. That would be a function of the healthcare kind of treatment context. That's just one example. And the other ones, I think we will probably have to dig in further in terms of like both the genetic and the healthcare kind of mechanisms that go into them.
Thank you. And let's see the question from Mateus Luis.
Yeah. Hi, everyone. I'd like to thank you all for the presentations. My mind is like full blown up with good ideas right now, so thank you all. I'm a Brazilian scientist in the beginning of my scientific career, so I don't know if this question is like dumb or anything, so my apologies. But Brazil has a huge, large data set of EHR, but we don't have as many genetic information as real-world data or EHR, which is, I think, common worldwide. But as Brazil has an mixed population, as Rachel mentioned, I think, I'd like to know from any of you who like to answer this, the inclusion of an admixed population on these type of studies that uses this approach of EHR combined with genetic data from a population that is admixed, that doesn't have an ethnicity or a defined race, is it going to be like more helpful to have a different population with more genetic traces and differences combined with the EHR data? Is going to help the precision, or is it going to be a factor that's going to make things more difficult to study in this population? I don't think if I made myself clear, so please, if you have any questions.
Thank you. Who would like to take this question? I mean, I think it would be both. It would be improving precision medicine, and it would also be difficult. But I think there are efforts to incorporate and better understand admixed populations, and there's a lot of progress done in that. So yeah, that would be my quick answer. I don't know if anyone else wants to add to that.
Yeah, I mean, that's Maria's exactly right. Admixed populations do make genetic analysis more complicated, but we can't afford to throw out a substantial proportion of the world's population. We need to use methods, genetics methods and phenotyping methods that can conduct analyses in admixed individuals. So I don't know if anyone else has anything to add to that, but I guess it makes it complicated, but it's definitely still worth doing.
Right. And there's a growing set of tools, right, to do this kind of research. And the Latin American Genomics Consortium does have a specific working group working on building these pipelines so that it's easier for all to appropriately analyze these data, right? So the point is to just reduce effects from population stratification. But I agree exactly with Rachel and Maria that from a precision medicine point of view, some of the extra analyses and things like that, it's worth it ethically and scientifically.
Thank you. And do you think at this point that we are advanced on this type of research and this type of approach, is it something that would be right now, as in 2023 speaking, like possible to be studied on at mixed populations right now? Or do you think we need more advance on specific ethnicities and race so we can export to at mixed populations? Or is this something that we can already make an effort and put on to start working in at mixed populations?
Yeah, so first of all, I'd just like to comment a little bit on sort of at least our definition of race, ethnicity, and ancestry. So when we're talking about like biological factors, the reason why we're concerned with ancestry is because we want to avoid population stratification, right? We don't want to report results that are just due to artifact. When we're talking about race and ethnicity, that's in the space of are there sort of social determinants of health that are impacting disease? But right now, currently, there are analyses underway, genetic analyses in admixed populations from Latin America. So it is possible. 2023, we can do this work, and we can do it robustly.
All right, thank you.
Thank you. And I want to thank everyone for joining us, and I'd like to thank our wonderful presenters and the PGC for inviting us, and wish you all have a good rest of your day. Bye.
Thanks, folks.